Thank you for the introduction, and thank you for the opportunity to share our updates in 48-Hour Discovery. 48-Hour Discovery is a Canadian biotechnology company incorporated in Edmonton 7 years ago in 2017. And our business model is provision of early discovery leads for precision oncology, radiopharmaceuticals, and other classes of peptide therapeutics. How many of you have heard of peptides? Any show of hands? Okay, great. So I can probably skip the introduction about the peptides, but just for clarity, peptides are a unique class of molecules that occupy chemical space between traditional small molecule therapeutics and classical biological therapeutics such as antibodies. And probably one of the most famous peptides today is Ozempic, which is a short peptide equipped with chemical modifications. So there's been in recent decades a surge in interest in drug lead candidates based on peptide modalities. And since 2017, 48-Hour Discovery has been employing its cutting-edge peptide discovery platform to provide leads for pharmaceutical companies interested in developing therapeutic entities based on peptides. And our primary interest and primary business model was in enabling the area of so-called targeted radiopharmaceutical drugs. These are the type of drugs that bring radioactive isotopes to the tissues of interest, oftentimes cancerous tissues, and eliminating them by focusing the radiation through delivery. We worked with a number of global biopharmaceutical companies, including top 10 therapeutic and pharmaceutical companies. And last year, we decided to start our own therapeutics portfolio. And in addition to assisting pharmaceutical companies in developing peptide-based therapeutics, we started our own pipeline in internal development. This is a bit of a nominal announcement here, but if this is part of the ongoing effort of 48-Hour Discovery to close the pre-seed round in an amount near $10 million. So it's going to be a very, very non-technical presentation. And if you would like to see a little bit more, we have a fairly well-developed website that goes in the details about how this platform technology works. So I do invite you to visit it. And if there are any inquiries that you have, please leave your comments there. I'll have only one technical slide to describe the process in general, and simply to emphasize that we operate in the drug discovery space, but we're not a virtual screening company. We are a wet lab company, so to say. So we like to handle real molecules in quantities. This is not too many zeros. It's right. It's about the right number of zeros. So our freezer contains about 100 billion chemical compounds, and it's a pretty small freezer. And in fact, only the top shelf of the freezer contains these compounds. These compounds are labeled with DNA tags, and they're effectively stored in about 12 different vials. So 12 different vials contain 100 billion compounds. Each compound is tagged with a unique tag composed of DNA. So rather than storing compounds in flasks with the paper label made with a Sharpie, we store compounds on the surface of bacteriophages with the DNA tag inside of these bacteriophages. And those of you who might have heard of technology called phage display, it's kind of a third generation derivative of the classical Nobel Prize winning phage display technology, in which translationally made peptides are displayed on the surface of bacteriophage, and information about these entities is stored in a DNA bacteriophage. What we add to this field is ability to combine chemical and biological diversity So chemical diversity comes from our ability to introduce post-translational modifications into translationally made peptides. And hence, we can operate with diversities that seem astronomical to chemists, but are fairly easily to handle biologically. And in a way, we take available biological diversity and make it more drug-like. This biological DNA encoded diversity is amenable to very traditional discovery pipeline through selection, affinity maturation, and directed evolution. Very much the same techniques that would be used to develop biological drugs, but in this case, we apply them to discover chemically upgraded peptide modality. And a typical endpoint of such discovery campaign would be provision of a handful between 10 to 100 chemically synthesized compounds to the pharma partners and licensing these compounds for the future preclinical development. So we're not certainly the only company in the world that makes the provision of peptide-based modalities for drug discovery, but we are perhaps the only company in the world that combines all the benefits that chemical and biological diversity has to offer. So again, we're specifically in the area of this so-called radiopharmaceutical technology, where we would like to deliver a radioactive metal such as lutetium encased in a chemical cage called a chelator, and use a peptidic structure to specifically target a unique type of tumor. There are already 2 FDA-approved modalities on the market targeting neuroendocrine tumors and prostate tumors, and they target unique receptor sites, and we would like to address all the remaining tumor modalities by developing specific peptide structures that will be targeted to particular receptors on unique tumor sites. We're the only company in the world that can build these billion-scale complexity with everything already in place. Targeting handle that contains biological diversity, chemical linkers that provide optimal stability and pharmacokinetic properties, and this metal-containing cage that contains the radioactive metal, all built together, attached to a vessel and encoded by DNA. And what I like to call this, it's the one test tube that contains all potential future radiopharmaceutical modalities. They just need to be fished out of this mixture and discovered. So it gives us this benefit over existing companies such as PeptiDream Bicycle Therapeutics, Orbit, created by labs that operate strictly within fairly low diversity molecular complexity that does not contain these chelator entities. This built-in TRP element offering is unique in the world and not available to anybody else. And it has several layers of patent protection, including some issued patents in ability to synthesize, characterize, and encode these entities by DNA. At least 2 of these patents have been already issued a few years ago. The company has been growing more or less organically in the last 7 years, and the revenue has been steadily doubling every year. This year, we're projecting to have between $4 to $5 million in sales. And in the previous years, we had $3, $2, and $1 million respectively. These are confirmed revenue from the sales to the pharmaceutical clients. The business model is such that we receive a certain amount of money upfront upon signing, and there's a so-called biobucks, as people like to call them, or cumulative milestones for any entities that go into preclinical, clinical, and commercial. And the graph shows the cumulative milestone potential of the company is around $100 million as of December 2024. We have proven track record in dealing with major pharmaceutical companies, but I'm actually very proud of being part of growing ecosystem in targeted radiopharmaceuticals, where we signed several partnerships with companies such as Point, RayzeBio, Fusion, and Oranomed. 3 out of these 4 companies have been acquired for anywhere between $1 to $4 billion. And this happened only within the last 12 months. And all of them have been working with us prior to acquisition. or unmet, one of our existing clients, has recently announced a very substantial deal with Sanofi and $300 million. So we're proud to be part of this amazing growing ecosystem, and again, facilitate medical advances for companies that pushing forward radiopharmaceuticals as the new modality in oncology management. Oh, here's another technical slide. I'm sorry. I forgot about this one. So this technical slide shows our internal pipeline that discloses second-in-class imaging agents for mapping PD-L1 status in human patients. This is still a preclinical asset. We're planning to push it into IND-enabling studies early next year, but it originates from this vast scatter plot of dots where each dot represents a unique chemical entity, and the blue dots here are the compounds that specifically came from a DNA-encoded discovery against PD-L1 receptor, and other rainbow-colored areas represent other type of compounds from other pipelines that do not bind to PD-L1. Vertical axis here is strength of binding to the PD-L1 on the surface of cells. So the higher it is, the stronger these compounds bind. And kind of a very quick tour de force showing how many compounds we handle in parallel. It's about 12,000 of them at the same time being tested at the same time with live cells expressing PD-L1 receptors. And then within these areas, there's a quite a nice structure-activity relationship. So our medicinal chemistry happens all here. We don't have many chemists synthesizing compounds. About a few thousand compounds here contain all the necessary information that allow us to build the initial asset and optimize it from the micromolar to mid-nanomolar to picomolar efficacy. The compounds engage with the cells. They match in preclinical performance to first-in-class PD-L1 imaging agents from BMS. And again, as I said, we're looking forward to pushing them into preclinical studies and hopefully IND-enabling studies somewhere next year. This is part of the internal pipeline. So PD-L1 is kind of nearing the end of the lead optimization. EpCAM, yet another one of the radiopharmaceutical targets, is finishing heat to lead and entering lead optimization. And we have a series of yet undisclosed targets in early development, somewhere in the mid to low nanomolar range as confirmed hits. So we're hoping to expand this clinical program to include more assets and prioritize some of the existing assets for aggressive preclinical development. We partner with the number of prominent people on campus in the city, and Greg Chan was former member of RaiseBio, one of the first multi-billion-dollar acquisitions in radiopharmaceuticals. Edmonton is actually an amazing ecosystem for radiopharmaceuticals development. The radio imaging has been here for several decades, and was this third or fourth wave of target radiopharmaceuticals, we're hoping to maintain Edmonton on the world map of provision of TRPs. So with the help of Frank Woost and Freimet, we're hoping to initiate the clinical program sometime soon. And these are the closing slides showing that I'm a leading peptide scientist. So it's very kind for the business development people to put all these nice compliments for me. Thank you, Adam. I'm very proud to pass the reins to Rick Finnegan. I used to be a CEO of this company for the first 5 years. A year and a half ago, I stepped down and I recruited Rick, and he truly enabled everything, and these multi-million-dollar revenues and clinical programs have been done under his fearless leadership. So I think this is it for today. And again, as I said, if you have any further questions, and if you forgot the email addresses, 48hoursdiscovery.com, contact us, let's get in touch and talk about pretty much anything.